So I'd like to thank the organizers, specifically Ilya, for inviting me, and the funders. And today I'll talk to you about a specific subset of methylation sites and why I think they're very interesting. And if you heard my talk last year, we're going to expand that into causality and reversibility. So our journey starts with this one site. We were analyzing some methylation data in kidney, and you get a list. And at the list, you have the top of the list. This was the strongest correlation between methylation level and kidney filtration rate. And you get a nice p-value, nothing unexpected here. But looking at this site, we've noticed it has intermediate levels of methylation in kidney, and it is almost completely methylated in every other tissue. And this is something you don't see a lot. So we asked, is this a coincidence? We gathered all sites that look like this, 253 of them, and all sites that look like the mirror image of it, because why not? And every single site, without an exception, regressed to the mean upon the progression of kidney disease. Highly, highly statistically significant. And you can say, well, Danny, no, it's actually regressing towards 0.5. You're just running away to the middle. No, it's not. If you look at sites at the higher end of the distribution, methylation levels of 0.7, it still goes up. And vice versa, 0.3, it still goes down. So these sites, they don't regress to 0.5 or any other arbitrary number. They go to where the rest of the tissues are. And we interpret this as loss of epigenetic information. You have a very unique signature for that tissue that deteriorates and becomes more like everything else upon disease progression. What is this good for? So if I take the information from these sites and I just throw it into a PCA plot, very, very simple. Just do a PCA plot. Here are my patients. These are the sick people. I don't know anything about kidney. I don't know anything about kidney disease. I haven't characterized these sites in any way. I just throw the data into a PCA, and there are my patients standing out. And again, the more unique the site is, the stronger it correlates to, for example, fibrosis in kidney. Again, very significant p-value. Do they capture any of the biology of the disease? So we can take these sites and look at where they sit and what DNA is around them. And they're highly enriched for transcription factors that are important for kidney development and are important in kidney disease. And genes that are associated with these sites change upon kidney disease. Okay, so that's nice, but that's one disease. What happens in other diseases? So we went on and we collected all the sites that look like this. And if you get 100% hit rate, your criteria is probably a bit too harsh. So we relaxed the criteria a bit. We wanted a 0.1 difference from the 5th percentile of 30 tissues compared to that. And I hate to introduce new terminologies, but I have to because our lists are very different than tissue-specific lists described previously. So we call this unique low and unique high because it has a unique pattern in that tissue. And we collected sites that look like this. Many tissues have several thousands of them. And the most important thing, the coloring. So if it's UL, it's blue. If it's UH, it's red. We'll keep this scheme throughout this presentation. I'm not showing here placenta. Placenta has more of them than any other tissue, but we don't care about it here. And if you look at these sites, so in different tissues, they're enriched for different transcription factors. They all have their own unique signatures. Okay, so we had kidney disease. What about liver disease? We collected data sets of multiple liver diseases. We clustered them into one. Sorry, we didn't have the resolution to do this one by one. And as you can immediately see, the UL sites, almost all of them showed a positive correlation to liver disease. and almost all of the UH sites showed a negative correlation. If based on my kidney data, I had to guess here, I would get 99.6% right on a few thousand sites. To do the p-value here, you cannot do it in Excel. You have to go to R and tell it to do a p-value in log scale. Otherwise, you don't get a number. So, but if I take the same sites in the same people and look what happens when I do adipose tissue, I get nothing. I get a small distribution across 0. So information is lost only in the sick organ. Kidney disease, all kidney. Liver disease, old liver. Nothing happens in adjacent tissues. What about other forms of stress? So when we are exposed to sun, this is very healthy. Go out, spend time in the sun, but your skin will age. It's called photoaging. And this predominantly happens in the epidermis, in the outer layer, and much, much less so in the dermis. So we can take data of sun-exposed and sun-protected sites, and again, using skin sites and unique methylation sites, you just throw it on a PCA, you see the nice separation between dermis and epidermis, and the nice separation between sun-exposed and sun-protected. You don't see here a nice separation between young and old. These are the small circles versus the big circles. But sun-exposed and sun-protected naturally segregate. And this does not happen if you do any other subset of sites. You do random sites, you do sites with a high standard deviation or low standard deviation. You don't get this nice separation. So epigenetic information is lost upon stress. Another way to visualize it, so we can look at the difference between the pan tissue average and our site. And if we'll get red on top and blue on the bottom, it's the opposite of what we've seen previously. This means we have a regression to the mean, what we call information loss. If they're mixed, we get nothing. If we get it the other way around, we have divergence, which I won't know how to explain. And as you can see, liver, like you've seen previously, red on top, blue on bottom. Our control, and reviewers had a really hard time getting this. This is a control. This is where I expect it to be mixed. Autism is not age-associated disease. We don't expect to see any information loss. And indeed, we see no information loss. Some of the blues and the reds are above and below the line. Everything is mixed, just like we expect. If we look at adipose tissue in type 2 diabetes, red above the line, blue below. If we look at sun exposure epidermis, red predominantly above, blue predominantly below. But if we look at the pancreas in type 2 diabetes and the dermis in sun exposure, we actually see divergence. We don't know how to explain this. We haven't found a rule to watch which tissue behaves which way, but we've seen it in other cases as well. Another mystery for now. What is the functional importance of these sites? Well, running 15 chromatin states against 100 different cell lines here for the liver samples, as you can see, only one thing pops up really strongly. The UL sites are tissue-specific enhancers. They are enhancers, they have all the markers of enhancers, but only in that tissue. And you can say, well, Danny, what you're seeing is actually changes of cell populations that are associated with disease and aging. In some cases, to some extent, this will be true, but this does not explain this completely. We see it in places where we don't see significant changes in cell population, like aging brains. And we also see it in purified single cell populations. Here, for example, CD4 cells, as you can see, very strong regression to the mean upon aging. So this happens not only with cell populations, but also within the cell. Okay, we wanted to validate these findings. We took 2 more studies. And again, as you can see, hypomethylation correlated nicely with UH, hypermethylation correlated with UL. very, very highly significantly enriched versus controls, which is just random sites of the same group size. Not only that, we took another study where we had fibrosis grade in the liver, and the grade of fibrosis correlated with the amount of epigenetic information loss. So we can quantify how much information is lost, and we get a nice correlation with the grade of fibrosis. So information loss really identifies the disease and captures its severity. Well, correlations are nice, but what about causality? And if you heard my talk last year, causality is hard. That's the hard part to do, especially because we're working in humans. So one thing we can do is we can take known SNPs to be pathogenic for liver disease and see, do we see an overlap? So 17 SNPs were selected, and for 7 of them, we saw a strong enrichment for liver unique sites. The expected value was 1. So we see a strong enrichment for pathogenic SNPs. We can also go and look at sites identified by Mendelian randomization to be either adaptive or damaging for aging. And we took here 2 criteria, aging GIP1 and longevity 19th percentile. So basically, you have either things that contribute to aging or adaptive response to aging that come to mitigate it. So UH sites and only UH sites show high enrichment for damaging in both criteria. UL sites, and this is again general aging, we don't have a site list like that specifically for liver. I wish we did. Damaging shows a mild depletion. We don't know if it's statistically significant for UL. And in mice, if you go and you look at liver clocks, liver aging clocks, again, we see a very, very strong enrichment for these lists. Is it reversible? Can we actually turn back the clock? So we collaborated with the lab of Ido Goldstein, and they're doing alternative day feeding in mice, basically intermittent fasting in mice. You feed them day yes, day no. They established very robust protocols, both in mice and in cell culture. And what we saw was that if we do alternative day fasting, we see very, very high enrichment in areas that were changed with our liver unique sites. A single fast will do nothing for you. If you want to fast, you have to maintain it for a while. We saw regression to the, we saw a divergence, okay? So going back to the youthful state upon alternative day fasting, and this was, most of the sites showed divergence, only a small percentage of the sites showed a regression to the mean. Single fast, again, did nothing. And again, this is not the results of changes of cell populations within the liver, because if we take hepatocytes, grow them in culture, and treat them with hormones that mimic the effects of dietary restriction here or alternative day fasting, we get, again, very, very strong enrichment for liver unique sites. So upon fasting, the liver of the mouse, we know it regains much of its function. We know from works of Ido Goldstein and others that specific primers and enhancers are primed. We also see that epigenetic methylation that was lost is regained. We're going back to a more youthful state. And then, I'm not much of a liver expert, Ido is. He chose a bunch of transcription factors. His students did the analysis, giving them our UL sites. Every single one of them, specifically in UL, showed very, very strong enrichment within these sites. Again, supporting the expected physiological roles. So to summarize, we show a very strong support for the epigenetic information theory of aging. This is a mode of analysis you can apply to multiple public data sets. It's very robust. Epigenetic information loss is in an organ-specific manner for multiple diseases. This is not true for all diseases, but it's especially true for age-associated diseases. From the standpoint of information loss, aging is a form of disease, but more so, multiple diseases are just aging of that specific organ. And what I'm showing here today for the first time is that epigenetic information loss is both causal and reversible, at least to some extent. And we hypothesize that closure of tissue-specific enhancers drives smoothing of the epigenome, aging, and disease. And this is basically our model. You have disease or aging, you see regression to the mean, and the sick organ becomes more and more like all the rest of the organs in the body and less like its unique signature. For UL, this is enhancers. UH story is a bit more complicated. We have stronger evidence for this to happen in age. UL with age, we have less strong evidence, but it does happen with disease. And this is reversible, at least in liver by fasting. Thank you very much for listening. And this is mostly the work of Naor and Roni, and thanks to Ido Goldstein and his students for collaborating, and I'll be happy to take questions.
I apologize if I missed this in the talk, but can you attribute some of that dynamics to cell types, to de-differentiation perhaps of some of the cell types? Is there understanding at that level? Thank you.
So the cell types we looked at, you don't see strong de-differentiation just from the nature of the cells, so I don't think it could be some of the explanation in some of the cases, but in many cases it's not. It's really cells kind of managing to regain their more youthful state.
I realized that in the neurons, you don't see this separation. And I was wondering is this because the neurons are so heterogeneous? There are so many types of neurons, more than probably any other organ.
So in neurons, you see the phenomenon more strongly in bulk tissue. You do see some regression to the mean in neurons. Glia actually work the other way around. They kind of diverge away. It could be. It could be that we need to really work with pure cultures and we need to be a lot more careful. Also, a lot of times they go and they sort the brains to generate this data and they just say neuron glia. It's a very crude way to sort neurons. So yeah, that could be it.
Yes, please.
Is there a technique to, like a biological method to try and methylate or demethylate a specific site, and then you can see like how it affects a specific cell type?
So you can today with tools go at specific sites. I wouldn't want to do this because I think the power here is you activate programs that work on thousands of sites in parallel, maybe even more. And I think working on a single site here is not that interesting, and I wouldn't expect in most cases a very strong phenotype. Trying to inhibit or methylation or demethylation globally to try and address the roles of causality of epigenetics and try to give a number to how much is it is actually something that's very interesting to us, but we haven't done it yet.
Yeah, I think we are one brief question, brief answer, and we will have to move on.
Sorry for maybe stupid question. Am I right that you will observe the same loss of information if cells undergo cancer transformation, like EMT, so if they become in cancer?
So I always try to avoid cancer because it's such a big field and I know so little about it. I think in cancer, and again, it's 100 different types and it may be different for different ones. I think you'll see a movement that isn't as directional as we see here. I think you'll see something that's a lot more mix of some gain and some lose.
Thank you very much, Danny. I'm sure you will be happy to discuss further during the break.